let me make an announcement. Okay. Let me make an announcement to everybody here that next week there's going to be, I think, a slightly different Zoom address to go to, because next week we're having our bias panel and we're going to run that through the Zoom webinar software rather than just the regular Zoom software. So I sent that out to the whole class yesterday through an email, through Canvas. So make sure that you notice that that's a different site than the one we're on today. So hopefully by announcing it now and sending you an email, we won't have too many people going to the wrong site next week. And that's really just for next week, because it's a panel, and when you have a panel, it gets more difficult to control it. Okay, so welcome everyone. My name is Wes Hines. I'm the department head here at the University of Tennessee. This is our weekly Colloquium program. We're happy that you're all joining us. Today, we're very fortunate to have Dr. Ken Brooks give a presentation. He's the Vice President for Sales, Project Management, and Corporate Strategy for Intra-Opt Medical, where his efforts have focused on image guidance for electron radiotherapy, IGRT, designing and launching a dermatology LINAC optimized for in-office treatment for non-melanoma skin cancer, and introducing and selling Flash preclinical versions of the system with the intent to add Flash as a clinical treatment option available on both platforms in the future. He earned his Bachelor of Science in Nuclear Engineering from UT in '87, so he's one of our alums. And he has a MS in nuclear from MIT in 1989. He returned to formal education a decade later, was formally educated in medical health physics and radiological engineering, completing his PhD at Georgia Tech/Emory University program in 1993. He has a very interesting background. He served as an assistant professor at Emory, a vice president at Varian, and even spent some time early in his career at NRC. So Ken, welcome.
Thank you, Wes. Thanks for having me. And I'm glad to be speaking to the Ball Nation. Go Balls. So as you said, my agenda today will be talking about Flash, or bringing Flash to you, but it'll be covering a little bit of background of our company and where we are, and then Flash developments at large, what's going on in the country and in the world, and then what are we doing specifically with Flash. So what the W's, what is it, who's doing it, what are they doing now, and so on. And hopefully we'll have time at the end for a discussion, and you can have a lively question and answer session. So company Interop is a privately held startup company in Sunnyvale. The founders were formerly from the radiotherapy divisions of Varian and Siemens. And in fact, those control systems originally were designed off of the Siemens design. And it's ironic that those two companies have now just merged their radiotherapy, and Siemens has just bought Varian. So it's funny, that's kind of comes full circle. But the founders left those companies and formed Interop about 20 years ago. And they spent 2 decades commercializing what was then a new technology, was X-band, high frequency, small compact accelerators for generating MEV electrons, and getting evidence for a multitude of disease sites for intraoperative radiotherapy. And you'll see that if you're not familiar with this field, that acronym IORT, we'll use it a lot because it signifies a whole product line, our whole original product line that we started with here at the company. And now we're in the 3rd generation of Mobitron. It's a mobile device. It rolls into the operating room. There's videos of it on our website, of both it and the derm version. And the derm version is stationary. It goes into dermatologist's office. It uses the same particle beams. It just uses it stationary. So that's kind of the 2 product lines that we have. And now I've come in, as you mentioned, in the last couple of years since I've been here, I've come in and cranked up the machines, and just basically cranked them all the way up, maximized the output, and lo and behold, the systems were had a lot of overhead to expand to, and we've been able to get Flash to work very well with it. And so, what is Flash? Why do Flash? Flash represents the rare chance to expand the therapeutic window in radiation therapy. So, what do I mean by therapeutic window? Well, that's really the difference simply between normal tissue dose tolerance and tumor kill rate. So the wider you can make that, the more effective the therapy is going to be. And so really, if you look at the last 30 or 40 years of radiotherapy with external beam, It's been very sophisticated, conformal, and so on. And that's gone about as far as it can go. It's done a lot of great things, but Flash looks like it's the chance to take it to the next level. Ultimately, we feel like Flash could impact millions and millions of patients. I'll show you that in a second. And both of our product lines could possibly use it. We also think that we don't think that low energy, a lot of folks in the industry might say, well, this is just a 6 or a 9 MeV electron beam. It can't go very deep. But there are enough lesions at shallow or intermediate locations to really control this rollout in the R&D phase, investigational phase, so you can figure out what strategies to adopt for deep lesions. I think going after deep lesions, we'll show you in a second, with protons or other things, are very, very difficult and challenging. They can be done, it's just very difficult to do. And I think sort of playing around with Flash on this low energy regime lets you inform yourself what to do for VHEE, which is another acronym that stands for very high energy electrons, and that's electrons over 100 MeV, protons and MeV photons. So that's kind of the overview of the company. And as I mentioned, the company has spent 20 years developing intraoperative radiotherapy, and you may not realize this, but advanced diseases listed here on the pie chart represent half of its breast. The other is colorectal, pancreatic cancers, head and neck, very nasty cancers that you don't know what to do with. Maybe they failed primary therapy. You can come in with intraoperative radiotherapy and give them something else to consider to try to extend their life or save their life. And so we've grown the business to be expanded in these indications. And now, of course, we've added skin in. So skin is essentially 5 times that. So if you look at the cases treated in normal, traditional, variant Linacs, Electrolinacs, so on, so around 1 million patients a year in North America. The big green Pac-Man looking chart at the bottom left here is 5.5 million derm lesions treated a year. Mostly, if you see on the right pie chart, mostly Mohs surgery and ablation from dermatologists in their offices. But there is some electron therapy shown there, and that is really done mainly in large bunkers in hospitals, traditional hospitals. And so you can see there's this missing element of having a small compact electron linac that could fit into a dermatologist office. So that's what we've added to our portfolio, and then possibly adding Flash to that. So that this really opens us up to a large amount of patients, 5 times the number we were facing with IORT alone. So we feel like it's a good strategy going forward. And we've been successful. We've got about 100 sites now around the world in 17 countries. And these are marquee names that we can't even fit all the good names on here. Places that have been long-term users. Stanford was one of our first users there in the neighborhood there, out of the factory, and places all over the world. IMRT, and now they're beginning to be users of derm and Flash, and that's really, really exciting to see. So that's kind of the background of the company. So look, what's happening at Flash Development Europe? First of all, what is Flash? That's ultra high dose rate radiation delivered at dose rates between 40 and 1,000 grays. It's reported all throughout the literature. The total dose, however, remains 8 to 30 gray, the typical kind of total course of dose you would get, maybe over 30 fractions. Now it's in 1 fraction. So the total beam on time to be able to do that kind of a total dose at that dose rate is milliseconds or less. The first human was now treated at the tube in 19, is published in 20, and this is the publication on the right. And you can see this was a lesion on the upper forearm of the patient here near the elbow. And it was a 3.5 centimeter resistant disease. The patients had multiple of these lesions all over their body. And so it was ethical to treat them, and they got clearance to treat them with the 5.6 MeV electron beam from an industrial linac, not our linac. However, they have now acquired our linac, but they use an industrial system for this. 3.5 centimeter field of view. They used a 5 millimeter bolus, 90% coverage, so it treated about 12 cc's of tissue at that energy. They used 10 pulses with 1 microsecond pulse width at 100 hertz, so it was 90 millisecond duration. So that was an effective average dose rate of 160 grays a second, or 1.6 grays per pulse. And you can see in the picture there and the results, that's the 1A figure shows the original simulation 3 weeks in, and then 5 months in. And at the bottom, you can see what the paper doesn't say. On the front part of the forearm, they treated 2 other lesions at the normal 10 gray a minute that every linac in the world is calibrated to. And it left scarring and clear side effects from the 10 gray a minute trying to treat the same kind of dose they did quickly. And so the Flash is essentially shows no sign of ever being treated, whereas also it was an ulcerated lesion before treatment. So the flash seems to have a durable effect. That patient's now 2.5 years disease-free there on that arm, and I think we're going to have a picture coming out very soon that shows that even better picture than that. And the flash seems to be durable. So what's it doing? What is flash doing to tissue to cause this to happen? How can you do dose rate that fast, that much dose at one time, and not leave scarring like you saw on the top of the forearm? It's because it is apparently inducing what's called transient, and I mean very, very transient, very, very short transient hypoxia by providing a protective effect to normal tissue. For pulse sources, the average dose rate, total time, and instantaneous dose rate, it's very dependent upon that. So your total dose, as well as all these other factors of the pulse structure itself, because these are pulsed electron sources, that really matters. And so there's a lot of research that needs to happen to go in to figure out what does it, maybe it can vary with disease type or cell type, maybe it can vary location of the body and so on. So far, the flash effect, this transient hypoxic effect, has been seen and demonstrated in multiple organ systems, brain, CNS, intestines, lungs, skin. These are mice. The only human ever treated so far is just this one arm of this one patient in the world. Fish, but all species have been also demonstrated this, from fish to mice to pigs, cats, dogs, and now humans. So the big thing about flash is, and I've mentioned therapeutic window earlier, is that you're giving this dose really high, and normal tissue is essentially not seeing the side effect. So that's a huge therapeutic window advance. And so that's because for whatever reason, while the normal tissue doesn't see the effect, the tumor tissue dies at the same rate, or even maybe it's more effective. And so it's a very interesting, shall we say, interesting phenomenon that's been discovered, and it's taken radiotherapy by storm. In my 25 or 30 years, I've never seen anything that's gotten radiotherapy so stirred up as this has. It's also now been shown and published in all major modalities that are used in radiation therapy now: photons, electrons, and protons. So we think it's here to stay, and it's a very interesting development in our field. And it helps to get a picture. You might be asking, how did we get to this point? You know, why wasn't this sort of thought about or discovered before? Well, it turns out 30 years ago or 50 years ago, people were doing radiolysis experiments, radiating water really fast and looking at the hydroperoxides that come off of water, or free radicals that come out when you dissociate the bonds from the radiation, and trying to correlate that to what it might do to DNA. But they never really took it to the macromolecule or the macroorganism. They just sort of, it was radiation chemists doing their studies and so on. They found some early evidence, like this rat tail study where they were radiating rat tails and some of them fell off and some of them didn't, and stuff like that. But it just seems like no one ever, solved the puzzle and went into applying it to humans till 40 years later. The groups in France and Switzerland did, and they started applying it to mice, lungs, guts, brains, pigs, cats, very sensitive tissues, and seeing this very strong effect. Then, as I mentioned, they treated the first human, and now from 2020 going forward, I think you're going to see the first trials announced in 2020. I think very shortly, our partner that treated that first human in Switzerland will also announce the first human trial using our machine. So as I mentioned, these are kind of the papers, and they're all in the last 5 years. Notice the data on these are all in the last 3 years, really. Mice lungs, brains, zebrafish embryos. There's a clinical vet trial going for squamous cell cancer of cats' noses, and cats get this squamous cell disease that if they let it progress, they can't smell their food and they quit eating and they starve to death. And so it's ethical to treat the cats, and the cats do miraculously well. They have a little loss of hair for a while, but it grows back, and they actually survive this very well, very high dose rates. Pig skin the same way. Pig skin very closely mimics human skin. So, low normal tissue toxicity, ISO efficient, or even better efficient, even more effective tumor control. How do you explain this biologically? And it's the largest factor that I think, it's really hard. It's like asking a bunch of people who politics almost. It's like, what is the biggest factor at work here? I think the largest factor at work is really flash producing less of these reactive oxygen species. So those folks back in the 70s were trying to measure that. And flash produces less of them. And so it changes this whole dynamics of this microsecond, picosecond kind of time frame, what's going on, where the differential between flash and conventional doses matter. And there's an effect going on, and these groups around the world doing flash are focusing on this very thing right now to try and come up with models that explain it. This figure is from Charles Lamoli. He's at UC Irvine. He's just got our machine, and he's working closely with a group in Switzerland who treated that first human, and he's been done a lot of animal experiments there. And so they'll both have our machine, have the same platform, and they will hopefully get a very much clearer idea of what's actually going on. But to quote Charlie, and I talked to Charlie recently, he said, tell people, everyone asks me what's going on and we can't explain it. We just observe this effect, and it's just like aspirin. People didn't know for 50 years why aspirin worked until later. And that's kind of where we are with this. So just sit tight and we'll give you the full explanation later. This is another slide from Charlie, and their publications are listed here, just showing you how the kinetics of the flash pulses proceed in time. And on the y-axis, you've got organic, the hydroperoxides that come off from a flash. Normal tissue, what happens in normal tissue, what happens in tumor tissue is quite different. And that's what's leading to this therapeutic window difference. And that's sort of been measured and published now recently as well. And people are after this flash, understanding and discovering this flash effect. I don't know if my video is showing here or not, but it should be animated. But there's two different pulse structures that you're going to see for electrons and proton beams. Photon beams are going to be continuous. So the photon beams that would be treating flash that are being proposed, and the one that was treated in Grenoble at the synchrotron light source was not pulse. So it was just solid on. So the two that really have a pulse structure are the one on the left for electrons and the one on the right for protons. And you can see that geometrically they're scanning a pulse, a spot scanning at some frequency on the protons. And so as soon as it leaves a spot, you're stopping the radiating, you're stopping the formation of these flash products and so on. And so the dynamics of that are very different than the dynamics of the electron beam on the left. So on the left, you see we have a repeated structure, or a pulse structure, or trains sometimes called, where you have a quiet period, a pulse and a quiet period, a pulse and a quiet period. That's just repeated 10, 20, 30 hertz, 50 hertz, 60 hertz. Our machine can go to 90 hertz. And so normally the flash treatment will be done in 1 to 10 pulses at like 60 to 90 hertz kind of thing. That's kind of similar to what was done for that first human. That's where we expect the human treatments, once they finally do get approved, to be in that range. This is animating, and you can see the animation of that. So you can imagine that the full volume is being pulsed with the radiant energy, the radiant particles, the electrons on the left, or entire volume of being hit every pulse, whereas on the right, just that little circle, the little pulse spot scanning size is being radiated. So protons have it's a lot more complicated to try and get a volume of any significant size. And you see that in the literature where they're just still treating small mice. We'll show that in a second. So here's that pulse structure I was talking about. This is an example here. 30 gray going to be delivered in our machine. We've taken films with this, shot films with this right here. The pulse is set to 4 microseconds wide, and there's a 16,663 microsecond space and time after that till the next pulse comes along, and we repeat that 3 times. So we can dial up. We can actually pulse level control our system and give you pulse level control, and we can let you vary the pulse repetition frequency or the gap between, and we can let you vary the width of the pulse itself. So we feel like the tool is very flexible to allow these laboratories and researchers to look at all these possible effects by conveniently adjusting the machine very easily. I'll show you that in a second how it looks. That whole delivery time, though, for 30 gray is 50 milliseconds. So the mean dose rate is 600 grays a second. And that seems insane compared to a regular linear accelerator, which is a fraction of a gray per second. It's 0.01 grays per second or something like that. Factor of 1,000 or 10,000 higher. The spot scanning proton delivery, this is from a recent article in 2020. This is UPenn's group. They're using an IBA system on the left here. And you can see what they have is they have a little, it's very hard, maybe it's hard to see on the picture, but they have a little a BB in there, a little lead shot that they scatter. So instead of having the spot scan around, like I showed you before, they put the mouse in there on the turntable, and they scatter that proton beam out so that it covers the whole region, like you see on the right over there. See the isodose boxes where it's covering the abdomen of the mouse, or two different packages there. You can see that. That's also an issue because they don't want to move the spot around inside the mouse, so they want to radiate. They want a solid beam of solid fluence of proton particles in the mouse. So that's kind of where they are compared to having that whole mouse. We could have 2 mice and our whole field could be 10 by 10 cm, and just the whole mouse could get irradiated. So there's a very big difference in the way this and the electrons work. But nonetheless, both Varian and IBA have introduced flash onto their platforms, their clinical platforms. They've announced upgradability to them. What you're seeing published so far is little cubes of 2 by 2 by 2 millimeters, or 5 by 5 by 5 is the largest I've heard of and seen by either vendor. They also have issues about whether to treat in a Bragg peak or not, and then they have range effect, range uncertainty effects, and so on. So it's a whole lot of complicated issues to worry about, I think, in protons before you're going to get to figure out what to optimally do in protons for humans. So we do think that because of that, the electrons and our research electrons will be the first thing will be this year announcement for human trials. So the group at Lausanne, this is Dr. Boris and his group over there, have tried to put together a chart of what it would take. Let me just sort of rack and stack where I see flash and where I don't see flash, and where is it coming in. And so this chart shows all particles, photons, protons, and electrons. And most of this in the group together in that top there, 3 to 11, is kind of like what we've seen with flash results have occurred. And then some of these are flash results don't occur. And then you can see the little red dots is our interop system, where it's going to fit, where it fits on the chart. This shows it a little bit better. You can see kind of what we think. So the idea here on the y-axis is the total time to deliver 10 grays, and on the bottom is the dose rate. So really what you want to be is, you want to be down and to the right. That's better to be down and to the right on this curve. And so the one pulse Febitron from the 70s, that was the radiolysis experimenting machine. That's something you couldn't do scale to humans or to trials in animals, but it worked great in water samples. And that had the highest, the most optimal pulse structure for flash generation and creation. So we feel very well suited here on the chart of what's being done around the world and with different particles. So it's interesting to see where the protons are over there on the left side of that, and then their dose rate, effective dose rate, is lower. I think they can get the dose rate higher, but they have to do, again, many more complicated things to get the dose rate higher, and it's never to the whole volume. So I do think it's got some technical hurdles ahead of it. So really, where does it stand? Well, as you can kind of infer from my discussion so far, it's still more questions than answers, but progress is really rapid. The major issues to study are the modalities. You know, which modality to look at, and what are you trying to do? What are how do you optimize these parameters? The beam structure I just described briefly for the protons and the electrons. It's very complicated to do. You need equipment in labs to do that. And then once you know that, you got to update your models for clinical application, and you got to maybe figure out how they work with systemic agents or immunotherapeutic agents. There's a lot of people thinking maybe there's an immune response going on here as well. So all of that has to be looked at. There are major issues. I think everyone's tackling those things, and everyone's wanting to seek this chance to get into this new area of research. Really is the potential to reposition radiotherapy in its fight with cancer. Radiotherapy is kind of the last option of resort compared to surgery and chemo. It's the third leg of the stool for cancer therapy, and this is a chance to bring it more mainstream. It's really can expand the therapeutic window way beyond this intensity modulated radiotherapy, image guided radiotherapy, and stereotactic radiotherapy, IMRT, Linux, all that stuff is trying to expand the therapeutic window by geometric optimization, but it's not affecting the fundamental biology at all. Still the same fundamental 10 gray a minute delivery. And so this flash takes you to a point where that could be meaningfully looked at and changed and improved upon. You still might want to use the conformal, but if you could do it with flash, it would be better added to that. So it's going to take a concerted effort to collect this meaningful data. Lots of trials are being conceived of and thought of. We're selling machines to people who are doing that process right now. Multiple kind of basic science studies of radiation chemistry and radiation biology are proposed or underway. I do think the governmental agencies are already getting in. We're already seeing grants issued to Irvine. UC Irvine just got a PO1 grant for this and Charlie Lamoli I mentioned earlier, and that's helping him fund his efforts there. I do think you're going to see I think Wes is probably the UT guys and Oak Ridge guys, maybe can get him on some DOE activities in this regard. So I think that the government probably has the scale to explore some of this. There's a lot of work to explore, a lot of things to study, and it can have a meaningful impact, I think, not only for medical radiation therapy, but for things like space travel. If you're trying to go to Mars and you're worried about radiation damage or radiation dose and so on, I think having this kind of understanding of the model at this point at this time scale may be useful as well in those activities. So I do think has a lot of potential for future growth. As I mentioned, here's Professor Boris that I quoted his charts there. You see him showing you on his very chart where he's pointing to where he would like a machine to be, which is almost impossible to get a machine to be there. But we're moving in the correct direction. He has the majority. I mean, these guys he came from France. He came from that group in France doing it. And he's put together a world-class team here. There's our machine in the background. You can see sitting there. It's just gotten rolled in. That's the day that it rolled in. That's earlier this year. So during COVID, we're still shipping machines, and Flash does not slow down for COVID. They're looking at, they've really done everything only with electrons. And they plan to announce human trials here very, very shortly. You'll see announcements very shortly on that. That's the first site in the world. The first site in North America is Wes's alma mater in Ohio State. And they have a machine. That was just a few weeks ago. They took a picture of a machine under testing for commissioning and so on, calibration. This group was very interested in a lot of very interesting applications. I should have a picture of the veterinarian up here because the vet school is interested in this as the radiation oncologist is for humans. And so this group is really set up to do it great. They have large patient populations with recurrent advanced disease, and they would love to begin to work in trials for Flash. They're already doing some trials for us in breast and recurrent head and neck disease. So we really are pleased to have Ohio State as the first in North America. I don't have a slide for it, but the second in North America is going to be McGill University in Toronto. That machine has shipped, and that's just in the process of being installed. And I'll have a slide ready on that very soon. That's in process of being calibrated and commissioned. So the third in North America is now McGill in Canada. And then you can imagine that the other names are lining up. I've got several to announce in the next few weeks, which we'll send announcements out on that. And you'll see some really big names come into the fold here for people wanting to test this device. So what is it people are looking for in interop? And I've given a little bit of background of what the big players, IBA and Varian, are doing in protons. Elekta is the other big player in radiotherapy, and they don't have a Flash play right now. They're not. I think they're just watching and waiting to see what's happening. If anything, they may be partnered with IBA more, and that's kind of their toe in the water for Flash. So that's really all there is in Flash right now, except for some research companies who make research clinics. So what is it that we're doing with our commercial system at Interop Flash? And this water tank here is showing you that there you see the blue flash, and this is slowed down to where you can see the flash, but that's a 9 MeV beam, and that's the first time the shrink off light can show you what the actual shape of the shape of a 9 MeV beam of electrons looks like. And it's hard sometimes for lay people to understand the electrons actually stop, and they stop in the tissue. They don't keep going, which is why electrons work really well in IORT and why they work really well for Flash, because you limit, unlike photons, you may need to make sure you account for where the photons go as they go in transit through the body. So the electrons work really well, and you can see that displayed here in the water tank very well. We've done greater than 60 grays per second. I'll show you in a second. We've got up to 600 or more grays per second now in the current systems. It's 6 and 9 MeV. Our platform can go to 12. The waveguide is long enough to do 12 MeV. We're looking at a higher power source to get it to where we can maintain flash power at 12 MeV. And so that basically lets you, if you had 6, 9, and 12, that lets you have 2, 3, and 4 centimeter treatment depths. And that gets you through quite a lot of lesions, especially the ones we're talking about, IMRT surgical beds and dermatologic lesions. That's a good way to start, good place to start. You don't have to be doing CT scans and worrying about three-dimensional effects because it's a 2D problem with electrons, clinically, at least at these energies it is. And it's a mobile platform, so you can move it from place to place. You can move it out to get a different version. You can move it around. People can share it. And in fact, I'll show you in a second, we share it commercially with our clinical system. The system now, we're quoting 40 to 800 grays a second, and I think very shortly we'll be up over 1,000. Our goal is to get it to 2,000 grays a second and somewhere on the order of 15 gray a pulse to explore the bounds and limits of these flash treatments. We think, and the researchers think, we don't need to go much beyond that. Probably sort of the threshold for when you lose effectiveness is somewhere in that range. And right now our field sizes are up to 10 centimeters, and that's what you see here in the water tank, 10 centimeters. So as I mentioned, we have 3 configurations that are possible in our system. They're just simply 3 rows of options you have. One option is flash only. That's a dedicated flash and radiator. That's convenient. Multiple disciplines can use it. You can have it setting in a, like you could have it in the basement of a nuclear engineering program, for instance. You could be sitting there radiating samples, and then you could drive it, so you could get the mobile version or the stationary version. You could drive it somewhere else. In the bottom slide shows you the clinical IRT with flash. That has our motorized drive system. You have a joystick and you can drive it around. It'll go like half a mile or something on a battery charge. So it's designed, the head lays flat and it can go into elevators in hospitals and it's within the weight limit of the elevators, and you can move it around on campus or move it around in a medical campus or research campus and move from place to place. If you buy the clinical system, what we do to be able to maintain our regulatory certification is we have the flash cabinet, which is our control cabinet for the 3 channels, sold separately as an add-on. So if you buy, look at the top row, if you look at the flash only, that flash cabinet gives you 2 flash channels, 6 and 9 MeV, and 1 conventional channel. So that lets you have your standard back at 10. What I mean by that is 10 gray a minute. That's a normal dose rate in a conventional machine, and then 2 at these very high dose rates. So down at the bottom, you get the clinical system, you get the conventional cabinet, 6, 9, and 12, and you get the flash cabinet as you have in the top row. So that lets you basically share the expense and help pay for the research machine in a clinical research system, like you see at Ohio State, for instance, where they can use the machine clinically and then also use it for research. And so we have 2 different clinical platforms that I mentioned briefly before. We have the Scanable Derm platform, and then we have the IRT platform. And so you could just choose, if you're going to buy in one of these anyway, you could just add a relatively small amount to the order and get a flash controller, and you could be doing flash experiments with this system. So that's sort of the near-term goal. Intermediate term is to get these trials going and get regulatory clearance for superficial indications for both IRT and for derm. And then longer term, look at very high energy electrons, which is electrons over 50 MeV, where you can treat anywhere in the whole body. The thorax, you can reach anywhere in the body with that energy, or other technologies, protons, photons. A lot of people are arguing photons and coming up with a very interesting design. The phaser design from Stanford is very prominently known about, and that's a very promising design. I think that has a lot of merit in the future. So it's just a wait and see for the longer term. But I think in the intermediate term, this kind of platform people can find a way to pay for it. They can find a way to get their experiments done and use existing clinical platform that's already verified by the market for 20 years. The modifications we made to this clinical system, to this little flash controller that I keep showing with a little lightning bolt on it here, it's for standard system in center column, flash on the right, 6 and 9, 2 channels, 6 and 9 for flash, 1 channel you choose 6 or 9 for your conventional calibration standard. You can vary that pulse width and you can vary that frequency, and that sets that pulse structure up that I showed you a few slides back. And you have to flip 2 toggle switches. You have this little radio knob button here to change the pulse rate, frequency, and pulse width, and that sets that pulse structure for you. And then the control screen has been modified, and when you flip the toggle switches and you go into this controller, instead of seeing where you enter monitor units or enter dose, you enter number of pulses. So you just control by the number of pulses, and it calibrates and gives you that dose. So this is the way this research system is set up. We expect a consortium of a dozen or more sites working on this, and then as we understand and the people have experimented enough with knowing which ones to work, we'll tighten this up and make it to where you can also put in a dose using a certain given pulse structure. So we kind of do a reverse lookup and make the control system do that more convenient. But right now, it's all granularly broken out for you researchers to go in and play with every part of the pulse control system. So what we've done in taking some standard measurements to talk to people about it, we've come up with a series of distances. One at the point A is B, C, D, and E, and A is right at the exit window, the beryllium window of the LINAC, where the electrons emerge. That point is just a few millimeters off of the window. It's not actually flush against the window, but we have a bracket there, a mechanical bracket there. You can locate animals there and test fixtures there. You can locate film there, and that's how we measured it. That's a 4.5 or 5 cm exit window. That's how large the beam comes out of our machine at. So the blue and the red bubble charts here, the blue and red bubbles, are points A, B, C, D, and E, and the diameter correlates from going from 5 to 10 cm. So that sort of shows you how it varies with field size and dose. And then you see the dose rate there is 700 roughly, 668, 665 versus 600 for 6 and 9 at that point, at that exit window, point A. So people are doing experiments at different places, and you can kind of see, I have a relative meter points here for where some other systems were used or in other literature applications there with orthophone and variant, modified variant systems. So this is what we can do now. And this is well beyond, I mean, the human was treated at what, 1.2 gray per pulse. And that's our interop 9Es. You can see that's 1.2 gray per pulse at 10 cm. So we could have treated a lesion that big instead of a lesion that big at with that same characteristic beam and beam structure. These measurements were done also at 2 millimeters depth in solid water, 4 microsecond pulse width, and the 60 hertz pulse rate frequency. We've just now changed to a new higher power modulator cabinet power supply, and we can now go to 90. So these curves are going to shift up disproportionately from 60 to 90. So the whole thing will just shift up, and we haven't got that updated yet. We're in the process of measuring. But we continue to evolve this thing. We also continue to get enough power to have a 6, a 9, and a 12 as well. So you'll have all 3 different steps. So what QA and safety interlock considerations do we have in this current incarnation of the system? With the flash beam dosimetry, it hasn't really been modified. The exit window scattering pulse collimators have not been modified. So changes in the dose rate don't really change the PDD, the beam profiles, and output factors. So, and that's what we're seeing in measurements, and now our customers are seeing that as they do their own independent measurements. Do this high dose per pulse, you have to really look at appropriate dosimeters. You know, the monitor chambers inside the system that you use when you use a conventional mode, they're just capped on ion chambers. They don't work, they saturate, and you can't really rely on them. So you've got to come up with other ways of doing this. We're also working on our final version of what we're going to use for measuring the beam and sensing it to replace it in flash mode, the ion chambers that are there. But when you go back to conventional mode, we'll go back to the standard capped on ion chambers. That's in every LINAC system. So that's how we're doing the dosimeter now. We recommend, and our customers are using alanine. That's in the food irradiation business, uses a lot of alanine for testing in QA how much dose they're giving to food irradiation. TLDs, scintillators, radiochromatograms, everything's looked at and used. Ion chambers are still tough. We're working with the big ion chamber companies. We're trying to see if they can make an ion chamber that can be designed to tolerate this kind of a dose rate, and more on that in the future to come. But interesting area to study, very interesting area to study. Beam time limits. So one of the things we did is we made factory limits on beam time limits. And of course, in flash mode, when you toggle those switches up and put it in flash mode, you need to have some kind of a limit so people can't just let it run forever. So we put 200 pulses on the limit. That's factory. So that's our standard factory setting. We can negotiate that and make that whatever you'd like it to be. If you are a national laboratory, we can unleash it, and you could probably sign a waiver and you can have it run all the time, and it'll run day and night. At that, it'll sit there and run all the time and not overheat. We ran it. In fact, when we first started doing this, we turned it on. We didn't have the pulse control system turned on yet, and it ran for about 30 minutes, and then it melted the Kaplan ion chambers because they were still trying to measure charge. So we've got that to where you can basically turn this on and let it run continuously if you need to do that for some radiation hardening or other kinds of experiments. So it's more than just something that you could use for clinical work. You could put some minor changes here. We've set the duty cycle for so many minutes on, so many minutes off, just to protect everything and not have these things breaking everywhere. Although we're willing, of course, to work with somebody, like I said, to get it to run longer intervals if you. We don't really have any increased power needs. The power needs are pretty minimal. It's 220 volt power. It's 20 amps circuit, not much different than any other regular piece of medical equipment. The stray radiation also remains roughly the same. So the shielding requirements aren't that high, but your workload can be so high out of the system because it puts out dose so fast. We recommend, of course, and everyone's putting it in bunkers so that there isn't any concern about workload affecting the symmetry of the room it's in. Moving forward, the first 3 systems I mentioned are already installed, and they're at various stages of commissioning and testing. We're already getting some initial zebrafish results back saying they're getting a flash effect. 2 more systems are going to be delivered by year's end. We'll have announcements very soon of those. One of them is going to be UC Irvine, as I just mentioned, Charles Molly. Another one's a very, very big name in radiotherapy and oncology in general in North America. We're doing a collaboration with those sites. We've already identified 4 potential trials with these institutions, and 2 of those will be announced before the year's end. Those first 2 trials will be the CHUV and Lausanne, and I think others will come out very shortly behind that. we're in process for certifying our high dose rate mode functionality, and it operates in physics mode, so it's not clinical. It's not a clinical mode. You can't mistake it and use it on the clinical system at all. It's impossible to do that. You have to physically plug in the flash controller, flick the switches, and intentionally go into physics mode, which has a password. So you do have to get into physics mode, and that's being certified now. We're working with the countries of mentioned here, and to get an IDE, an equivalent in those countries, so that our clinical partners can begin doing experiments with humans. Right now, you don't need an IDE for animals. Technical roadmap is, as I've already mentioned, insinuated, we're increasing the power supply with a solid state modulator. We expect a significant increase in the dose rate and dose per pulse, as I say, up to around the 2,000 level. We're pushing 1,000 now, or maybe 800 or 900 now with this latest upgrade, and we think we can double that one more time. And that will get us beyond any kind of range of testing that might be needed or wanted in the industry. If it needs more than that, we can certainly look at more than that, but we don't think it will. None of our researchers think it will. And then, of course, our main focus now is getting into the engineering tasks of the control system for interlocks and QA, and thinking about how you put this together in terms of dose so that people can dial up the dose that they want to give to the project. So that's kind of where we're going in the roadmap and what's happening with our developments in the company. And just to close out here with you, there's a future topical events. If you're interested in this topic, there's a couple of really interesting things coming up in this month. In fact, there's a 3-day seminar that the SHUV, our partner there, and the CERN. Everyone knows where the CERN, about the CERN, I'm guessing, in nuclear engineering, where they're looking at VHEE and looking at ultra high dose rate. So it's basically VHEE flash, like I mentioned it earlier. You can go to this web link here. You can click this link, and I'm going to give this presentation to Wes. So Wes will have the PowerPoint slides. If any of you, by the way, if any of you want that, you're welcome to it. And go to this, register for this session. It's fully remote, and there's the times and when it begins. And I recommend anybody interested to go into that. And you'll see what the CERN's doing with VHE. You can see some of the long copper structures they have there to get to very high energy, 100 MeV and so on. And then we also have a flash webinar coming up. And that TUBE is, so the TUBE group that I showed in the photographs earlier, Dr. Boris, Dr. Mary Catherine Bozanen, and Dr. Moakley are all three going to be hosting a physics, biology, and radiation oncology viewpoint of flash at the TUBE with our machine and their experience. And so you can certainly, I'll send the invitation. We haven't got the invitations ready to go out yet. They'll be electronic. I think they're done by Zoom or Teams, one of those platforms. We'll do that webinar on. And so look for that if you want to join that. Feel free to join us on that. And that's really, that's my presentation. Wes, I'll pitch it back to you and we can.
Okay. Yeah, thanks, Dr. Brooks. So we'll go ahead and open it up for questions. Do we have anyone with questions?
So I've got one. First of all, the Terrain Golf Globe video was awesome. Thanks for showing that. I wondered if you'd please explain in a little bit more detail the nature of the differences in transient hypoxia that are induced in normal or tumor tissues. Is it free radical formation? Is it oxygen kinetics or oxygen fixation? Or could you explain?
Yeah, I mean, I'm certainly not a radiation chemist or radiation biologist, but I know a lot of them and I'm friends with a lot of them, and I have to tell you, they have a hard time explaining it. You can read these, they know what's going on, but it seems like the radiation biologists are saying, you radiation chemists had this 30 years ago and you didn't look at it. You didn't look at the biological aspects of it. So there's a lot of that going on. But the radiation biologists are the ones that have seen it 5 years ago in France and Lausanne. And this is a paper from that group. So this is Charles Lamoli's work here I'm showing now. And this shows the washout, the kinetics are different. And by the way, I think you're going to see them publish a group to further confuse the issue. They're going to further publish a group and they're going to say that while oxygen species, the fact that less reactive oxygen species are created is an important factor, it's not the only factor. And they've got some other things that they're going to publish that they're keeping confidential until it's published. So I think it's a multifactorial problem. It is the normal kinds of hydroperoxides, hydrogen peroxide, and all the list of hydroxy radicals that you get from radiation. It is all of those at play here. But which ones are direct action, which ones are indirect action, what's the ratio? Is it different with flash? Is it different with electrons and protons and so on? I think that's all got to be looked at. And the short answer is, I don't think anybody can tell you very well. I certainly can't. I know the references to go look at. When I read those references, I leave not knowing any more than I did when I read them, really, to answer that question. It's a very good question. I wish I could answer it for you, because if I could, I'd probably go for the Nobel Prize, but I don't think I know the answer. I don't think anybody else does either.
Thanks, Ken. Really interesting talk.
Thank you. You're welcome. Thanks, Jason. Did somebody else raise their hand there?
Yeah, I raised my hand. Actually, my question was very similar to what Jason asked. Thanks for your talk, Dr. Brooks. That was very interesting. Yeah, so this question of transient hypoxia is puzzling in a way. I'm wondering, do they know why a high dose rate would induce this transient effect, whereas a lower dose rate with conventional radiotherapy would not produce this effect?
It's something to do with this. It's something to do with the kinetics. If you model them on that time scale, it's the washout. So there was a paper released by Nebraska group looking at that, doing the kinetics, doing the traditional model of kinetics, and all the very adds and subtracts and the differential equations. And so there's a full paper on that. Actually, you can look at that, and they come up with the conclusion that it should be possible. So it's just strange that we scratch our head that nobody ever tried it before. But
so this paper that you have by Charlie here, is this a measurement or a model prediction?
I'm sorry, can you repeat that? Is it a what?
Is it a model prediction, or is it a measurement that they did?
It's a model. It's a model, but then they compare it to measurements at the end.
Yeah.
It's a model, and they say the model is in the ballpark of what you'd expect. It's one of those kind of things. It's what you'd expect early. Right now, not enough people have enough devices to even measure it. So if you look at the lion's share of all the publications are done by Boris and his group at Lausanne with an industrial attack at 5.6 MeV. I mean, that's it. It's one guy with one radiation biologist and one physicist.
Right.
The people in Switzerland, I'm not taking anything from Paris, it's maybe more people than that, but it's not much more than that. And so those two institutions are lion's share of all the publications. And now everybody else has jumped in and tried to quickly pour tons of money into it for protons. So as IBA, rightfully so, I would do the same thing. And they think it can help their proton businesses. But proton adds a whole other, I don't know how you study all these things all at once. It's just so many degrees of freedom. I don't know how you're going to get there.
Right.
That's
interesting.
These really smart group. They're very smart people working on it, and I have confidence in them. But I just think, boy, they've got a tough. As hard as it is in electrons, they got it doubly in protons to figure it out.
Thank you.
What's more? Keep more coming. I mean, every year it goes by now. This is our second year of playing with this, and we're out publicly talking about it and selling it. 2 years ago, it was just conceptual, and we really didn't know. We didn't even have these publications yet. And so now every year that goes by, it gets more clear. And I think another year we'll have some more answers to those questions. I share you're in the former questioners' questions. I just don't know how to answer them, fortunately.
Great. Thanks very much.
Hey, yeah, Ken, this is Wes. So I'm trying to wrap my arms around this. So if you really talk about protons and you want to increase the dose rate by, I don't know, let's say a factor of 100 or whatever, flash would be, that means you have to send 100 times as many protons down into it, because you don't change the energy of the protons. You're just
no. In fact, that's right. The whole other problem protons have is what energy do you pick? Because they're debating whether to treat the first experiments. They didn't know how to control the Bragg peak because they didn't want to mess with the energy with a range modifier, which is essentially just a block it goes through. And so they use the raw beam. So they fix the energy unless you can have one of those cyclotrons that modulates energy for you. And they leave that energy the same. You're right. And so the beauty of protons is that you don't have to do that. It can use that energy to get whatever depths it wants and get the Bragg peak kind of where it wants to be at the normal dose rates. For this, it's just harder. It's just really hard to do for protons. For us, it's very straightforward. For an electron beam, it's very straightforward to see how the whole volume is getting hit at once. Whatever your radiant influence is in that volume, the ballistics are, the whole volume's getting it. It's very different for protons. For scanning,
it just seems like
there used to be a scanning electron. So what's on the right? If we 50 years ago, there was a scanning electron machine out in the field, and so that's not restricted to just protons that scan as a spot, as pencil beam. So it's just not popular now, but electrons can be done the same way.
Yeah. So what's the difference? You showed the 3 different machines that you have. What's the difference in your IORT, your interoperative machine? What makes that one different?
That one is, the difference in these two is the collimation system on the end and the fact that you have a drive system to drive it around. Otherwise, they're identical.
Okay. So yeah, the one you can drive around, but I mean, if it's IORT, if you had the other one fixed in your operating room, it would be the same thing. So the collimator is different.
Yeah, in fact, we just sold that one that's fixed in the operating room, but with an IMRT nozzle on it. For
Okay.
They have a bunker built into their OR, and Salzburg wants that in their OR. So you really, I mean, this is what we typically sell, but we can also do Mr. Potato Head, and we can put the nose from one on the nose of the other.
Yeah. So the IOR, so the collimator, that just allows you to focus the beam on a different type of area. So if you're inside a person, you know, interoperating, and there's a tumor there that you want to fry, it gives you a different way to.
It does. And so we have on the IRT one, we have a sarcoma applicator that's big. So we can actually, you know, get these sarcomas in your leg, these Ewing sarcomas or whatever. The surgeon will go and remove them, and then you get really good limb sparing results. You don't have to lose your limb and be amputated. We can flash that. I mean, you can flash it. We do IRT now with it, and we scatter the beam out to a field like this that covers a man's calf. So your entire thigh or calf, the whole lesion can be sanitized from the surgical bed, and it's just wonderful. You get really, really good results, very strong results in sarcoma. So UCSF has 2 of these doing sarcomas all day long in San Francisco.
Okay.
So it's
So
you make the beam whatever you want it to be, shape, electron beam.
yeah. So I just have one last question, and I don't know if anybody else does, but how did you get, you know, permission to do one human trial? You know, a lot of times human trials are done several at a time. You know, you had one human trial, and if it worked so well, how come it's taking, you know, a couple years to be able to do a second person? I'm kind of wondering how that all happened.
That's a good question. Thank you for asking this. I wasn't clear enough because I was going through this in a hurry. This trial here, and I'm showing the arm of the patient. This was done by the PI, John Borish, you see his name there, and that's the whole group at the Shooting in Lausanne. That if you get this, download this paper, this is free to download. So if you download this paper, you can read it. But in their materials and methods, they use the PNV Alsen industrial accelerator. They know it's not that the Swiss medics and the medical authorities won't let you use an industrial device to treat humans in a regular fashion. They still won't allow that. And so he got a special approval to treat this one patient because this one patient happened to be his patient. He was treating. They were treating with everything at the tube, throwing everything at this patient, trying to get these lesions to go away. And external beam radiation was leaving bad scarring. Chemotherapy was chemo resistant. It was surgically resistant. It's T-cell lymphoma. It's not your usual basal and squamous. And so he made the argument and won, and they let him do this one. Only could do one, and voila. I mean, and everyone else is scratching their head. Left the whole rest of the radiotherapy world on their heels. All the big North American sites, none of them are even near having a device to treat humans. He's treated the first human already. So it is a big deal. And now he's used a platform like we have. We have the proper regulatory authority to have our regular clinical device there, and then have this add-on console that in physics mode, you're allowed to put tests. You're allowed to put non-cleared things in physics mode because that's what physicists do, is test them.
Right.
And so we're approved for that in Switzerland now.
So do you kind of go from, you know, we can use this in physics mode, and then to get permission to move that towards a human, then you have to like show, okay, we can use it for animals, and then for animals, then you can kind of get another approval, and then you can move towards humans. That's the process?
That's what the process is. And we think we can. Well, the group in Lausanne did all of the animal experiments already, so they got a well-oiled machine. They're going to stack it up. You're going to see the physics paper come out. You're going to see an animal paper come out. The trial announced, and humans already on it in the next 12 months.
Wow. Okay.
Moving.
A couple more questions. Does anybody else have a question?
I have a question. With the collimator design, I was wondering how you kind of controlled the X-ray production, or if that's just negligible in this case.
Excellent. Oh, I like this question. We have some physicists and nuclear engineers in the room. So as you know, charged particles, when they hit high Z materials, they create brimstone photons, and basically you make an X-ray tube. And so our entire system is made out of high density polymer, Delrin, and other high density plastics. But we just don't see it. In fact, we don't generate. We have the lowest electron stray radiation and scatter radiation of any device treating like this, any variant or any other LINAC. Our stray is so little because we use plastic parts basically for that very reason.
Okay, thank you.
So your collimators.
Sorry, we have less than 1% photon leakage in that system. Very, very, very low contamination, low photon contamination. There is some, and you can measure the brimstone tail and the phantom at the range of electrons and see it. But our spec is less than 1%.
So your collimators are made out of plastic?
Yep.
Okay.
They're steam sterilizable plastic. They can reuse the OR over and over again for the IORT wings. And the derm is contactless. So the derm is classically done where you line the patient up. And you go to our website, there's a nice derm video. And I have the entire first site in Florida basically captured in a cartoon video. So you can see the exact workflow. You can see the derm collimator work. And it's just awesome. It lets you spin full 360. It's got a laser rangefinder in it for giving you the distance. You can drive the patient right to that distance and have your setup distance. And then your calibration is to the physics calibration is to that. And then the physicist can sign off on, the doctor can sign off on, and you can treat. And they're treating 60, 65 a day in that clinic in Florida on this derm system. So it's a hot, you can't treat that many skin patients on a big, clunky, large format LINAC. It's just the big electron tree is metal. It creates, like the last questioner said, it creates all this photon stuff. They really don't use it very much in skin. But this derm dedicated system doesn't do hardly any stray. It's the least stray beam that you can use.
So do you know about how heavy the head is of this machine?
Yeah, the head alone is the head alone. If you take it just to pull the head itself off, it's a thick aluminum frame, which is pretty strong and structural itself. It's a little too big for what it needs. You can make it smaller than it is, but the head alone is somewhere on the order of 400 pounds to 500 pounds.
Okay.
Then the back cart contains the modulator and everything's in the modulator cabinet. Everything's in that. So all that's in the little rolling cart is a dose control circuitry to control it. And I took the picture off the back. I could show you the different components. And it's in our documentation, in our physics guide. So I can also send you that and explains all the components and where they are. But it's a usual suspects. It's a magnetron powered system. And the video, there's a YouTube video that I announced last year with my voice narrating it, so you can't mistake who's talking with the East Tennessee accent on YouTube. And so go to YouTube and type interop space flash, and you'll see our video. And I take the cover off and I walk you through the whole process. And I had our head of engineering walk you through the process because everybody asked that question. And so that's on YouTube as well for you to look at.
What kind of services does it take to operate it? What voltages, or does it take?
It's a 220 volt DAC, single phase, and it's 20 amps.
Oh, okay. It has all the cooling? Does it have special cooling, or
the waveguide is cooled, but it's a closed loop cooling system with fans down on the back side, so it doesn't need any chilled water in the room at all. It's
okay.
It doesn't really add any more ambient heat than a 2 kilowatt light bulb in the room.
Okay. Great. Other questions? Okay. Dr. Brooks, we'd really like to thank you for your time. We'll make sure we get this posted. And if anybody else has questions afterwards, I'm sure they can give you a call. Thank you very much.
We have the deck, so feel free to share our slides around and socialize it and ask me any questions. Anybody wants to ask me more questions offline, feel free. Thank you.
Best of luck to you and your company. Take care.